The Bioreactor Lab was performed by Eric Bailey, James Hempsey, and Kenneth Richards.
In this bioreactor lab, we cultured a particular strain of E. coli using a bioreactor and inoculated flask on a benchtop shaker. The objective of this experiment was to compare the kinetic parameters for bacterial growth using the LB media to those of the M9 media. We measured the bacterial optical density in a spectrophotometer over a span of 6 hours in order to construct a bacterial growth curve. Due to the lengthy nature of a single reactor run, we could only complete one reactor run during the lab period. The equations shown here govern the kinetic parameters for the growth of E. coli. X denotes biomass concentration, S denotes substrate concentration, Y sub X of S denotes the maintenance coefficient, which for this experiment was 0.5. Mu denotes the growth parameter, K sub S denotes the Monod constant, and mu max denotes the maximum growth rate parameter. Since we were working with E. coli, we had several safety precautions that needed to be followed. Gloves, goggles, and lab coats were worn at all times. If we were to walk away from the reactor bench, we first had to remove and dispose of our gloves and wash our hands with antibacterial soap. Gloves also had to be disposed of in a separate trash bag. All contaminated glassware was put into the wash basin, and gloves and bench tops were cleaned regularly with 70% ethanol to avoid contamination. All E. coli samples were put into a waste container as well. The day before the experiment, the bioreactor and all glassware to be used was cleaned with soap and water. We then autoclaved the reactor, glassware, and pipette tips at 120 degrees Celsius in order to sterilize it. We also autoclaved 3 liters of LB media at a ratio of 20 grams per liter of distilled water.
To make the LB media, we mixed 20 grams of LB broth with 1 liter of distilled water and shook until a homogeneous solution was achieved. The day before the reaction, the reactor was assembled and the DO and pH probes were put under UV light for sterilization. In addition, flasks of LB media were prepared for the inoculation of E. coli. After the DO probe had sat in the UV light for 15 to 20 minutes, it was then placed in the reactor so that it could polarize for 6 hours. The day of the experiment, the reactor was hooked up with the water lines, the DO probe, pH sensor, and thermocouple, and everything was calibrated. Then the E. coli was added to the reactor. The temperature was set at 37 degrees Celsius, and the impeller was set to 500 RPMs. The flasks with E. coli were put on the shaker, and the temperature was set to 37 degrees Celsius. Samples were taken every 30 minutes for 6 hours and analyzed using spectrophotometry to obtain their absorbance. After all the readings were taken, all the waste was discarded into the proper waste bins, and the reactor and glassware were cleaned thoroughly.
This plot shows E. coli growth in the benchtop shaker. The graph was made by taking the natural log of x over x naught versus time, which helped us to obtain the mu max. Dilutions were made to the sample after 212 minutes because the absorbance readings began to go over 1.0. A trend line was taken for the first 6 data points, which led to a mu max of 0.0181 reciprocal minutes. The next plot shows E. coli growth in the bioreactor. Dilutions did not have to be made for this circumstance. A trend line was taken for the first 6 data points as well, and the mu max resulted in being 0.012 reciprocal minutes. Using the M9 data, a 1 over mu versus 1 over s plot was created. Using the trend line, the mu max and the Ks, or Menou's constant, was found. Relating that trend line to the rearranged version of equation 3, mu max proved to be 0.0125 reciprocal minutes, and Ks, or Menou's constant, was 1.002 grams per liter. Compared to the results, the benchtop shaker promoted E. coli growth better than the bioreactor since it had a slightly higher mu max. The results also show that LB media may have had a more positive effect on E. coli growth than the M9 media. This is because the mu max in the LB medias were higher than the M9s. The samples were not collected when E. coli began to reach the death stage, which is why some of the results might be skewed.